Shall we start? Shall we start, or we should wait for a few more minutes?
We can go ahead and start, but
Okay.
I leave it up to you.
Okay. Let me share my screen. Okay, let's start. Welcome everyone for the statistical and AI methods for health data science seminars. Uh, those seminars are hosted by the Startup AI. Startup AI is a group aimed to organize activities that empower statisticians to participate more actively in AI research and leadership. It's dedicated to enhance the role of statisticians to addressing real-world challenges through AI research. It also emphasized the dual importance of leveraging both statistical methods and AI tools, ensuring that statisticians are not only participants but also influential leaders in applying those combined approaches to solve practical problems. Startup AI is also officially recognized as an ASA interest group. More details can be found by the website here. And this is the steering committees and the inaugural officers of the group. And this event is also co-hosted by the SAA sections on statistics in genomics and genetics. And you can scan the code here to get access to the previous seminar webinars. Okay, today our speaker is Dr. Michael Wu, and the title of the talk is Turning Crop Data into Scientific Discovery, Statistical Magic in Microbiome Science. Dr. Wu is a professor of biostatistics in Public Health Division, Fred Hutch Cancer Center. He is also a fellow of the American Statistical Association and former chair of the ASA/SSG. And he's also the winner of ASA Bob Riefenberg Award and ASA W.J. Yoden Award in interlaboratory testing. I believe many people are very familiar with his work, like Scott, Descartes, and Mercator. I believe he must be a cat person. Okay, and besides his success in research, he's also a black belt in Kung Fu and martial arts. So never mess up with him. And before the start of the talk, I also want to advise our next talk will be August 11 from 1 p.m. to 2 p.m. by Dr. Russell Shinohara. Okay, without further ado, Dr. Wu, please take your stage.
All right, thank you so much for the introduction. Let me just get to share screen. All right. I assume you can see?
Not now.
Oh,
I can see it.
can you see it?
Oh, yes. Yeah.
Okay, perfect. So thanks again for inviting me to be here. And I have to admit, this is probably one of the most challenging presentations that I've had to put together. In large part because I think the purpose of this is to primarily be somewhat didactic over here. So my goal really is to to help people who are less familiar with the human microbiome identify potential problems and potential inroads into starting to carry out human microbiome research from a statistical and analytic perspective over here. So as a result, it's a little bit of a hybrid presentation. But nonetheless, let's muddle through and see what we can do here. So let's first take that title apart over here. So as I was putting together, we can talk about crap data and statistical magic in a number of different ways. So in one view, what we're dealing with is data from crap or poop, which is where a lot of human microbiome samples are from. And from the other end of things, all right, you can view things, the statistical magic, as fantastic statistical analyses, fantastic analytical strategies that lead to important scientific discoveries. An alternative view, however, is crap data is terrible data of extremely poor quality. And statistical magic is when we describe analyses and tools as like magic. Oftentimes that's because we're talking about we have no idea why it's actually working, how it's working, and sometimes whether or not it's actually working. So that's oftentimes how we describe black boxes. And the truth is, both views are actually valid within the context of human microbiomes science. So here's an overview. I'm going to talk a little bit about the microbiome. I'm going to talk about standard analytic workflows. And then I'm going to dive into methods that are used across specific parts of the workflow. And then really, this is where I want to spend more time, talk about areas of opportunity, both including what sort of the current state of the art, as well as opportunities for more work in certain areas. So the human microbiome, all right, when we talk about the microbiome, we're talking about all of the microbes that colonize a person, all of the bugs the microbes that live in and on us. The vast majority of this is bacteria, and oftentimes that's what most people are talking about when they talk about the microbiome. But bear in mind that there's also a lot of other things that are constituents of the microbiome, things like virus, fungi, and other strange and wonderful things. Why are we interested in this? Well, it ends up that we have approximately as many microbes as human cells living in and on every one of us. We have about 10 times more microbes if you ignore red blood cells. And if you're looking at in terms of genetics, we actually have 100 times more microbial genes as compared to human genes. So in many ways, we're as much microbe, as much something else as we are ourselves. Now, the microbiome is found at many different body sites. For some time, we used to say all body sites, but that's sort of an evolving, that's something that's evolving. But the interesting thing is that the composition varies both by site and importantly by health status over here. So individuals who are sick are oftentimes, well, you'll see different microbiome profiles as compared to healthy individuals. And so here are some of the many discoveries that have already been found. So we found that the microbiome is related to all sorts of different types of environmental or generally defined exposures, things like diet, exercise, drugs, smoking, alcohol, as well as treatments such as immunotherapy, such as other types of pharmaceuticals and other types of interventions there. We've also found that the microbiome is related to outcomes. All right, things like asthma, cancer, diabetes, hypertension, and other health outcomes, both beneficial and harmful. Now, I put a little question mark over here because it's oftentimes not clear whether the microbiome is shifting because you have cancer or whether it's the microbiome that is conferring risk of cancer over here. A lot of times what we see are these cross-sectional studies, and it's very, very difficult, if not impossible, to tease apart sort of the directionality of the effect here. This is in contrast to human genetic studies where you know your genes are not being changed by outcomes over here. So let's dive into a couple of these so-called magical discoveries over here. So one of them was looking at topical tenofovir efficacy. So tenofovir is used for preventing HIV acquisition and treatment over here. And so the in the CAPRISA-04 trial, they compared topical tenofovir to a placebo, and they showed that HIV acquisition was reduced in the experimental arm by approximately 39%. However, there was considerable heterogeneity in response based on vaginal microbiome profiles. So if you look within patients or subjects who had lactose bacillus dominant vaginal microbiome communities, all right, you see that the efficacy was on the order of 61%. So this is generally associated with a healthy community. If you look at a non-lactobacillus dominant community, typically something that you think is less healthy, you see that the efficacy was only approximately 18% over here. And so what they did was, one of the nice things about bacteria is that you can actually culture them. all right, grow them in a petri dish to try and understand what's going on and try to look behind that so-called magic curtain and see what's happening. The major finding here was that Gardnerella vaginalis, which is a particular type of microbe, actually was leading to tenofovir depletion. In particular, this bacterium eats and metabolizes tenofovir. Consequently, when it's eating it and digesting it, then it's no longer able to do what it's supposed to do, including prevent HIV acquisition. So this is an example of where you have a very, very nice result that is highly supported by the science over here and with mechanistic things. Another area right now of considerable interest is in cancer immunotherapy. So we know that checkpoint blockade-based immunotherapies have had a major impact in cancer within the context of melanoma. You know, if you had late-stage melanoma, it was basically a death sentence. And response rates were very, very low over here. Currently, response rates are now on the order of 40 to 60% with modern checkpoint blockade. All right. And that's fantastic, but it ends up we have to realize 50 is much better than 5, but half of patients still have disease that is not responding to treatment. Now, where does the microbiome play a role? Well, in 2015, all right, some folks in Chicago found in using mouse models that the microbiome is related to response. This was reinforced by a number of major human studies in 2018 and more studies that continue on to this day. So overall, there's agreement that the microbiome in some way or another is related to sensitivity to immunotherapy or perhaps immunotherapy resistance. All right. However, the mechanisms underlying this remain very unclear. The two initial studies in 2018 found totally different bacteria to be related to response. And recent studies are still finding different things. All right. The vast majority of discussions on whether or not on how the mechanisms may be playing out are pretty squishy, and there's no systematic agreement across all groups and across the field on why this is working. All right. So in many ways, this is what we call magic, but the field continues to move on, and people are now interested in microbiome-based therapies. So one reason why we're very interested in the microbiome is that it's eminently modifiable. You can modify it traditionally using probiotics, prebiotics, or even antibiotics over here. And we'll talk about other modalities as we go along. So in terms of generating microbiome data, let's start talking about some of the nuts and bolts. What happens is you typically take a specimen, you extract your DNA, and then there's two major technologies that are used for generating microbiome data. The first is what we call 16S RNA gene sequencing, where you sequence a gene that is inside of the vast majority of bacteria. So that's a 16S gene over here. And all right, you sequence this gene. It's inside of the vast majority of bacteria, but there's certain regions of this gene that are highly variable. And so based on those highly variable regions, you're able to distinguish different types of bacteria from each other. All right, and we'll focus primarily on this technology in this presentation. From this, you get taxonomic information, which is basically the abundances of different types of named bacterial species or genera, or so on and so forth. The other technology is shotgun metagenomics. I'm not going to talk about that today, but from that, in principle, you also get taxonomic information, but you further get some information on functional profiles. All right, so we'll dive into this a little bit more. And so then this is your usual microbiome analysis workflow. All right, it's very, very small. We'll dive into these. You start from sample collection. All right, you do all sorts of strange, wonderful things. All right, statistics starts coming up around this area. And then this is where a lot of the more traditional statistical analyses happen. And you go down to generate biological insights. All right, various hypotheses, and hopefully write super impactful papers that help you get tenure. So, all right, how do we begin? You begin by sample collection. Very, very straightforward over here. All right, you just got to get specimens in some way or another. From there, you do DNA extraction and library preparation. All right. And this is all stuff that is typically transparent to most of us as computational researchers. Then what you do is you do sequencing. All right. So as I said, 16S or shotgun can be used. 16S is cheaper and is probably still continues to be more common, though shotgun metagenomics is now becoming more popular as well. From the sequencer, you get raw reads, all right, FASTQ files. You do some QC, you do various filterings. Again, just fairly standard pipelines for the most part, all right? And then we get to the part where there's actually some real statistics. At this point, what happens is you take the reads, all right, and essentially you cluster them. So these reads are basically small sequences where of particular regions of the 16S gene, all right, where you can map them to some sorts of references over here. So you cluster them based on sequence similarity, and things that are very, very highly similar, usually traditionally at approximately 97% similarity, you said that was probably something that would be at the species level. And so those are what we called operational taxonomic units, all right, OTUs. More recently, all right, we now do a sequence variance from Dada2 from Susan Holmes' group, which basically takes which is basically a much more rigorous statistical model for doing this that takes into account sequencing errors and likelihood of two things being unique or otherwise. This is very, very statistical in nature. However, for most people getting into the field, this is probably not the place to start. From there, you do taxonomic assignment, which then gives you this type of table. From here, what you have is a table where, in this case, each column is a separate subject, and each row over here is a different bacterium. So you could have you know that this one over here is bacteria, actinobacteria, all the way down to the specific species over here. And so you get this basically N by P table, which most statisticians and many data analysts are very, very happy to work with. These numbers over here represent the abundance, or you can think the amount, the quantity of these different types of bacteria that are present. All right. So let's talk a little bit about the data, microbiome data, taxon or taxa. This is a named unit of analysis. You can think of it as a species, a genus, a particular type of bacterium over here. And what you have are sequence reads or counts that quantify the taxon abundances. The data are high-ish dimensional insofar as there are potentially many taxa, not always. And the raw data are essentially counts over here. The data are highly zero inflated, though the reasons for zeros are sometimes unclear. They are very, very over dispersed with no really good distributions for characterizing them. And there's underlying biological structure and also very ill-behaved measurement error over here. So at this point, this is where a lot of times statisticians and downstream analysts focus get a data table from the core labs. And this is where some of the more traditional statistics takes place over here, right? And so we'll dive into a few of these different areas. So one thing to mention, all right, the microbiome actually represents a confluence, a lot of different analytic and quantitative fields, everything from microbial ecology, which focuses a lot on multivariate analysis, biostatistics, all right, computer science and informatics, as well as math, physics, and engineering. All right. So really the analyses you'll find are really a hodgepodge from all of these different types of fields, all forming up microbiome data science. And from this, the objective is to somehow or another let this data carry us towards helping public health, informing biology, or even clinical science. All right. Okay, so that's sort of the overview. We're going to dive into a few of the specific analytic strategies that are going on over here. First, we're going to talk about diversity analysis, which is something that is more unique to microbiome data as compared to other types of omics data. All right, so the first type of analysis that almost everybody does is alpha diversity analysis. Alpha diversity is basically your usual idea of diversity within an ecosystem or community. So if you look within a forest, all right, you can see that the diversity here basically is there's 3 different types of animals: alligators, pandas, and tigers. I don't know what world that's in, but we'll pretend that that's possible over here. And in the same way, each of us has our own community in our gut. All right, just this is instead of lions and tigers, bears, now you have different types of bacteria over here. And so alpha diversity is quantifying this concept of diversity within each person. All right. Note for now, diversity is not a good word. So in most of my grants, I've been calling it entropy or variability, but that's neither here nor there. So in terms of measuring alpha diversity, all right, typically standard measures include richness, where you're basically looking at the number of different unique taxa over here. So for example, the richness here would be three different types of animals, versus here the richness is only one type of species over here. The other thing is to look at evenness, how smooth or balanced the abundances are. So this one, everything is pretty even. All right, you've got 3 of each, but over here you can see it tends to be dominated by a particular species. And so from that, what you typically then get is, depending on how you want to do it, you quantify alpha diversity by getting a single index over here. All right, whether it's the number of unique observed species over here, Shannon index, inverse Simpson, all sorts of different things are used. But at the end of the day, it's just a univariate measurement. So afterwards, you just apply your standard tools that we all learn in kindergarten over here. All right. One thing to mention, however, in an area of active research, the sampling intensity does affect the measurements over here. Essentially, what we're talking about here is how many animals you're actually capturing when you look within a community. You're obviously not going to capture all of the animals within a forest. In the same way, you're not going to capture all of the bacteria within a particular community. Consequently, you're only seeing a small snapshot. And so the sampling intensity, all right, is basically how many sequences you're obtaining over here, how many animals, unique individual animals you're capturing. We also call that read depth or library size. And you can see that the library size actually affects the metrics that you're choosing to use over here. So alpha diversity and diversity does depend on how many animals you're trying to catch in this case. All right. So how do we deal with this? All right, this is typically viewed, the total number of sequence reads is typically viewed as a technical artifact over here. The most common strategy that is used is rarefaction, where you basically subsample reads such that every person now has the same number of reads over here. So you're evening out the sequencing, the intensity with which you're capturing species, different taxa over here. Other strategies that people have used, regressing out sequence depth, they try adjusting for it in analyses, or there's a very, very rich literature on estimating the total number of unobserved species or taxa over here. It's very, very rich fields, all of these, and none of them work perfectly over here, which is why there's still quite a bit of work to be done in this area. But subsampling is by far the most popular. All right. Unfortunately, subsampling is also highly controversial. The basic issue here is that there's certain assumptions underlying the randomness by which sequences are being ascertained, which don't really hold out over here. There are also some philosophical and practical reasons. However, at the end of the day, oftentimes it's better than not doing anything else, and so it still remains the standard over here. So the other types of analyses, beta diversity, here we're looking at cross ecosystem or cross sample variability. Operationally, this is a little bit more technical. What you end up with is a distance or dissimilarity between every pair of communities, pairs of samples. So you get a matrix, n by n matrix that measures dissimilarity between, say, your microbiome and my microbiome over here. All right, lots of different metrics for measuring dissimilarity. Some of them focus on the abundance versus others focus on presence absence. Others can bring in external information such as phylogeny and phylogenetic relationships, while others do not rely on this over here. All right, bear in mind that presence absent methods can still be sensitive to sequence depth, and so oftentimes people do rarefaction. Once you have this distance matrix, people do visualization, they do clustering, or they'll do association testing or include it in other types of analyses. So the idea over here, when you focus on beta diversity, you're now looking across samples. And so you're looking at variability in overall structures across different individuals over here, across different people. Each person has a community, and now you're looking at differences in the structure across people. And so we're still looking just at this tiny corner over here, but this is closely related to some of the other analyses. So the most popular thing that everybody does with this is they do visualization. This is what this is called principal coordinate analysis, PCOA, which is just a generalization of PCA. In statistics, we call this classical multidimensional scaling. They are the exact same thing, just different names. Operationally, you center your matrix such that you get now a matrix of similarities, and then your principal coordinates are just the eigenvectors of this similarity matrix. And so you can plot the principal coordinates against each other and see if you can find anything interesting visually. One thing to note is that in microbiome, the percentage of variability explained is oftentimes by the top principal coordinates is oftentimes extremely low, which is different than other fields. All right. In terms of testing, there's a lot of tools that we've borrowed from. The most popular tool is borrowed from ecology. Essentially, it treats microbiome as the outcome and then uses an ANOVA where distances are instead determined based on the distance matrix rather than based on Euclidean space. And then they do significance by permutation. An alternative is to borrow tools from kernel methods. All right, and so the meerkat approach, intrinsically, this assumes that the microbiome is the predictor, and effectively it's comparing similarity in outcomes to similarity in the microbiome profiles over here. And it allows for analytic p-value calculation, or you can use permutation. Ends up that these two approaches are equivalent, as Wei-Pan showed, under many situations, but there are certain niceties that nice aspects about using kernel approaches. All right. One thing to bear in mind, within the microbiome literature, they always talk about Permanova and some of these things as testing differences in means or differences in centroid locations. In practice, all of these methods are testing the global null, i.e., is there any association between these communities and the outcomes over here? So from there, I'm just going to touch on a couple of other topics, a couple of other things, particularly multivariate, particularly differential abundance of individual taxa over here. So this is the usual work scheme. You look at global diversity, you look visualization, and then now what we do is we look for individual bacterial taxa that are related to outcomes. All of the methods here have to deal with a lot of inherent nastiness. So this is looking at the proportional of zeros per subject. You can see that within a subject over here, 70%, on average, approximately 70% of the bacteria are not observed. If you look at across bacteria, you can see that the vast majority of bacteria are only observed a couple times within a study over here, right? So you have a lot of sparsity. Simultaneously, you have these pretty ugly distributions that are heavily over dispersed, but highly variable in terms of their distributions when you look across different bacterial taxa over here. And so your approaches have to take this into account. In terms of commonly used statistical approaches, this can include application of classical approaches, all right, where you just directly apply something like a t-test or Wilcoxon test. There are approaches that we borrow from other omics, primarily RNA-seq analysis, and there's these days tons of tailored statistical approaches over here. So in terms of classical statistical methods, typically what you do is you normalize the data, where normalization means you set the norm of the measurements from each sample to be the same. And in microbiome land, we call this total sum scaling, where at the end of the day, you get the relative proportion of a particular taxon within each individual. So they now become fractional over here. Lots of other transformations that are used. I'm not going to talk about them in detail. But at the end of the day, you get this transformed variable Zij over here, and for each taxon. So this is the jth taxon on the ith individual over here. And then once you do that, all right, to understand whether the Jth taxon is related to the outcome or not, what you do is we oftentimes log transform it if it's highly not, to stabilize the variance. And then you again use the tools that you've learned from kindergarten over here. Note that by convention, most of the tools in omics treat the omics features as the outcome. This actually doesn't really matter typically for association analysis, but it can affect interpretation. And there are some outcomes where you basically need the omics to be the predictors over here. We've borrowed tools from other omics, all right, namely things DESeq2 and EdgeR and Limma. So these guys over here are basically negative binomial regressions. Limma is sort of a linear model with empirical base to sort of stabilize variances there over here. So this is just negative binomial regression, right? There's also a ton of tailored approaches, all right? So inevitably, these guys all accommodate some features of the data while ignoring others. So examples of these are approaches that accommodate the ugly distributions, approaches that accommodate compositionality, and others that accommodate emergent issues over here that, for the most part, tend to be less commonly used. In terms of approaches accommodating distributions, people have basically come up with every possible parametric data model that you could use, whether it's negative binomial, beta binomial, or these zero inflated distributions over here. All right. And then here is one that we've developed that is non-parametric, but also that is non-parametric. But for the most part, these are just trying to take into account the different distributions. Other approaches try to accommodate compositionality. So compositionality results from the fact that the total sum scaling or rarefaction makes it such that the sum of the total counts for an individual is now fixed at a particular number. Consequently, if you change one taxon's abundance, if one taxon goes up in abundance, all of the others will necessarily go down as an artifact of having done this transformation. This is actually a common issue in lots of other omics, but it's typically ignored. People deal with it in the microbiome largely because we can, all right? So I always ask, does it really matter? I have to be honest, I've never really seen it make a huge difference in practice, but microbiome people are pretty obsessed with this. So I'm going to say, yes, you do have to deal with it. And the most popular approaches that deal with this are probably the ANCOM BC family of methods, Linda. And the idea behind these is that they directly estimate and remove biases resulting from compositionality under various assumptions over here. Okay, I'm not going to talk too much about these, but they're very popular and commonly used as well. How do you deal with zeros? All right, as I've said, the data are very, very sparse. The most standard approach right now is to add a small pseudo count, or you can try imputing them or using zero inflated models, as we and others have done. The issues over here, the choice of pseudo count can make a substantial difference in the results. And zero inflated models are somewhat unattractive because they're assuming that zeros are true zeros. So that's to say that the taxon is entirely not present. Consequently, a count of 0 versus 1,000 would be the same as 0 versus a tiny, tiny number. Whereas this tiny, tiny number is basically essentially the same thing as 0. All right, you're treating it in the same way as you would something that is very, very different than 0 over here. Recently, again, Lee has developed a nice approach where they treat the taxonomic data essentially as left-censored data over here. Then you can use survival models. So these are some of the different areas, different approaches used for individual taxon analysis. Realistically, however, it ends up that people have done tons of benchmarking studies, and every single approach, there's remarkably little overlap between different analysis approaches over here. So this study looked across 38 different data sets and showed that, hey, different tools get very, very different approaches. Here's another one which showed that it really depends on the data characteristics and how you choose to do your normalization. All right. The most sobering, however, is that many papers have shown that the vast majority of approaches for differential abundance analysis really do not control false discovery rates and type 1 error appropriately. All right, the reasons for this are varied. But some of it, a lot of it is that many of the tailored approaches rely very strongly on distributional assumptions. All right, they're very, very highly parametric, and violations of those parametric assumptions lead to type 1 error inflation over here. Okay. So following that, typically we do auxiliary analyses. So commonly conducted analyses include building prediction and classification models within the context of microbiome. People love random forests. People will do network analysis. This is also commonly conducted. In terms of network analyses, people typically do things like Spearman correlation. They do some correlations that have sort of been adjusted for compositionality, and they will just basically transform the data and directly apply the graphical lasso. So fairly rudimentary in terms of approach. So that was a 20-minute overview of how people basically do microbiome analyses. So this is where I really wanted to spend a little bit spend the remaining time, which is really looking for opportunities to make do impactful data science within the context of the microbiome. The reality is many areas could stand to see a lot of improvement over here. All right. However, the truth is also that a lot of these areas are already relatively mature. And even though the approaches could be improved, people already have their favorite or standard methods already. All right. And so it's going to be very, very hard to make an impact. An example of this, individual taxon analysis, really, you could do a lot better. We know that some of these approaches don't protect type 1 error. Some of them do. All right. But it's very, very hard, all right, in practice to demonstrate that a new method is truly better. As a result, all right, it's very, very hard to really make an impact over here. So I would not recommend that as a place to really dive in if you want to make impact. If you just want to publish statistical papers, all right, that's fine. But if you want to do something useful, that's not the place to go. All right. I think it's much easier to make an impact by engaging in areas that are relatively new, that are very underdeveloped, or where there's no dominant methods over there. And so when that's the case, I think it's much more easy to make impact. And of course, you want areas that are scientifically meaningful as well. So what are some areas which I see as being very, very open? All right. One is in data integration. All right. Within the context of data integration, I mean combining data sets across different studies. So horizontal data integration, where you get more microbiome studies, or vertical data integration, where you collect other omics data types. All right. Both of these are fairly rudimentary right now. Longitudinal designs, they're very, very popular. The methods there all suck right now. Non-bacterial data. People mostly focus on the human microbiome and when they talk about the human microbiome, they're primarily talking about bacteria, but there's actually lots of other types of entities forming the microbiome, all of which tend to be vastly understudied. And then the other things include clinical trials designs, where people are now interested in using treatment modalities that are based on the microbiome over here, or that entirely microbially based. And there's been very little consideration given to the fact that this is a different treatment modality as compared to traditional things in cancer or cardiovascular disease. And there's others that I'll touch on as well. In terms of horizontal data integration, basically you're trying to get more combined data sets to boost sample size and power. All right. So within GWAS, you typically just do some sort of meta-analysis. Within the context of microbiome, that's really hard because most studies use different analytic methods, particularly within the context of individual taxon analysis. So by far the most commonly used strategy for horizontal data integration now is just data pooling, where you take your studies and pool them together. In the world of microbiome, they call this meta-analysis, but the reality is we know that this is actually mega-analysis. All right, so if you have 10 studies and you want to pool them together, what's the challenge? All right, well, it ends up that there's a lot of heterogeneity. You have effects from the fact that these are oftentimes from different populations. All right, there's different specimen collection and preservation protocols. You're doing different types of amplification and sequencing different parts of the 16S gene, as well as different bioinformatics and pre-processing. We can typically deal with the different bioinformatics and pre-processing by going back to the raw data, but the rest of these guys over here, you're sort of stuck with over in this case. right? So how do people deal with this? Options for analyzing pooled data, you can do nothing. Some people do that. You can adjust for your study as a covariate, which is highly limiting, or you can do batch removal, where you're trying to remove the study effects and scrub your data over here. So here's an example of PICOA plots from a particular, from the HIV Reanalysis Consortium. So here they pooled data from 10 different studies over here. Each of the different colors corresponds to a different study. Each of these points over here is a different subject over here. And you can see that there's tremendous differences across study over here. And so that's really, really problematic if you just want to pool the data, the raw data. So what an option for doing this is to do batch correction to facilitate integration over here. So the main idea is that you want to treat the different studies as different batches and then remove the corresponding heterogeneity in this case. One thing to bear in mind, differences in studies typically overwhelm traditional batch effects, which are really just talking about, well, this sample was collected a little bit later than that one, but otherwise processed in the same way. In terms of the major tools that are used for batch correction, popular tools include the muffin approach, partial least squares discriminant analysis, and Conquer, which is an approach that we've developed, at least in our benchmarking and some other groups' benchmarkings. Conquer seems to do well. I'm not going to go into too many of the details over here, other than to say it is a popular approach over here. That's based on Combat, which was developed primarily for microarray data. The idea behind it is very simple. You're just regressing out the effect of batch. A little bit more to it than that, but essentially you're just regressing out the batch effects over here, all right? Combat-seq, they generalize this to count data for RNA-seq, and here they put a distributional assumption on there, and then they remove the batch effect by, and within this context, they estimate the batch effect, and then they remove it. They again regress it out within a negative binomial regression. All right, Conquer is essentially the same thing, except operationally it uses quantile regression. All right, it's same idea as Combat-seq, but instead of negative binomial regression, it just uses quantile regression across a fine grid of quantiles. It uses a two-part model, so to deal with the zeros, and it's quantile-based to ensure robustness to distributions over here. So here are some of the squishy details. I'm not going to go into this over here, and instead I'm going to focus on what happens when you apply it to data. So here, all right, the HIV Reanalysis Consortium pooled data from 10 different studies. So altogether, there were about 606 taxa on 572 individuals from 10 different studies. The 10 studies were treated as batches containing 24 to 135 patients over here. And the key variable of interest over here was HIV status. Other covariates here included age, BMI, and gender. If you do a batch correction, so these are the raw data. This is what happens if you apply Combat-seq. Helps, but doesn't really do a lot. You still see a lot of differences between the different studies. If you apply Conquer, you see that it actually gets everybody to align relatively well. It is not perfectly on top of each other, but it does look quite a bit better over here. If you look at the R squared, so the proportion, the variability in the microbiome, that is explained by the study effect. All right, in the raw data, it's approximately 30% of the variability is explained by study. If you apply Combat-seq, that drops to 20%. If you apply Conquer, that drops down to about 1%. That sounds great. However, and it does seem to help a lot. However, if you look at the amount of variability explained by HIV status, all right, it's still the R squared over here is still only 0.69%. So the batch effects and study effects over here are still dominating the variable of interest. So there's a lot of room for new and better methods. I'm not quite sure how to approach this in a better way, but hopefully one of you or somebody else out there can come up with something. Lots of room for improvement for horizontal data integration. Vertical data integration refers to collecting more omics on the same studies. All right. So recently I took a look and asked, well, how many studies are actually collecting microbiome and other omics? And the answer is a lot. So I screened a bunch of papers for whether they're collecting other omics from some of these relatively good journals over here. And you can see that altogether, a little bit over half of them also conduct other omics profiling at the same time as microbiome profiling over here. Okay. What other types of omics data are being collected? The vast majority of this is proteomics, and then other and small molecules, as well as other nucleic acids over here. But protein over here is actually highly diverse. It refers both to specific individual proteins as well as targeted proteins, as well as untargeted mass spec over here. And so if you break this out, all right, you can see that small molecules is primarily metabolomics, and that is by far the most commonly collected type of other omics data over here. Other things include transcriptomics. Proteomics actually now drops to third, fourth, and sixth over here once you break it out by different types of proteomic technologies over here. All right, so essentially over here, we have a whole bunch of different types of omics data. How are we going to try and carry out the analyses? Well, before we talk about the analysis, you have to ask, what is the scientific objective? If you look at these hundreds of papers, you see that there's actually a few different types of objectives. One is to understand the relationship among the different omics features. So what's the relationship between the microbiome and the metabolome? Others are really trying to understand an outcome. So what is the relationship between the microbiome, the metabolome, as relates to HIV? All right. And then within the context of looking at outcomes, you can have a facilitative or directive analysis where you're using one omics layer to help the analysis of the other omics layer. So are you using the metabolome to help interpret the microbiome's effect or vice versa? The other way is to treat both omics layers on equal footing, and you're saying, hey, I'm really interested in both of these data types simultaneously in this case. In terms of what we see out there, all right, the vast majority of them are not just interested in looking at relationships among features. So only a small proportion of those were. Most of them were concerned with looking at outcomes in this case. Of these, all right, the vast majority of them, again, were treating them as balanced simultaneously, though there were many studies where you're using one omics layer to interrogate the other over here. All right. So what were some of these objectives? All right. And what are some of the methods that are used? Analysis methods largely fell into a number of different categories. This included, and quite commonly, just direct application of classical statistical methods. All right. They used very classical ecological or multivariate analyses, things like PICOA, Permanova, approaches borrowed from other omics, as well as mathematical modeling approaches and highly tailored approaches in this case. So tremendous heterogeneity in the analytic approaches used. All right. Some examples of what people are doing using multi-omics to understand feature relationships. They were doing things like mapping the relationship between metabolites and microbes over here. Tools that they used include basic correlations, okay, co-abundance networks or mantle tests. People were looking at relationship between microbes and gene expression. They did things like canonical correlation analysis, partial least squares, and also enrichment analyses. So lots of different tools being used. Things where they were using one omics data type to understand the other's effect on an outcome. So for example, here they were interested in whether there was a mediation effect over here. And so they did things like so examples include direct application of two-step Mendelian randomization in this case, or things like non-parametric mediation analysis over here. So a lot of different tools can be used to enable study of one on the other. In terms of balanced approaches, this was very, very heterogeneous. So in one case, for example, where people were interested in identifying a prognostic signature over here or a diagnostic signature, all right, what some people did was they just stacked the data types together, whereas others tried to combine them in a more using multivariate analysis and then do classification based on that combination. So overall, very, very heterogeneous over here. So in terms of opportunities for data integration, horizontal data integration, where you're looking at more studies, all right, Data pooling is the current state of the art. It really is, it's great, but it still is far from perfect. True meta-analysis has not really been done very much within this field. So there's, again, a lot of opportunities to do methods development there. In terms of vertical data integration, all right, a lot of studies are collecting multi-omics. There's a lot of different analytic tools that are being used over here, which is great in the sense which is a great opportunity in the sense that there's no set standard. Consequently, that means that there's an opportunity here to help develop approaches that are going to be widely used. Other areas of opportunity include longitudinal studies. We know that the microbiome changes over time, so longitudinal collection is becoming very, very common over here. But overall, and very generally speaking, the methods for longitudinal microbiome analysis are extremely underdeveloped. Approaches that work well for beta diversity analysis and paired analyses tend to be relatively rudimentary. Once you do longitudinal analysis, you can start looking at volatility, not much in that area. In terms of visualization, nobody really knows there's no systematically good ways of visualizing longitudinal microbiome data at this time that I know of. And for individual taxon analysis over here, all right, there's, again, a lot of room for development in this case. You can use transformations and classical tests. All right, there's some tailored work that make very strong distributional assumptions. And at the end of the day, many of these only accommodate very simplistic designs. They allowed you to add like a single random intercept or something. So there's a lot of room for building up more sophisticated approaches here. And then within the context of longitudinal analysis, all right, how you then do data integration, multi-omics analysis, network analysis, or prediction, all of these guys still are very, very open areas. Another thing that I wanted to very briefly touch on is dealing with non-bacterial data. So most of the time when we talk about the microbiome, for the most part, we're talking about the bacteriome over here. But other things that you can look at include fungus over here, which tends actually to be relatively straightforward, but not very well studied. And there's not been a lot of integration of these data, particularly with bacterial data. The other area that is becoming more popular is the virome, and the NIH just started the human virome program. And here they're looking primarily at viruses that are infecting bacteria, because that's the vast majority, perhaps 98% of the viruses that live within us. It's not COVID, it's not the flu, it's actually phage, things that infect bacteria. All right. And so we're now able to capture a lot and quantify a lot of these viruses, all right? But there's almost no tailored downstream analytic tools here. So this is another area of tremendous opportunity. This is a field that is extremely nascent, in which there's literally no tools that are truly tailored for this over here. All right, so I'm not going to belabor this too much, and I'm going to stop in the next minute or so. But I also wanted to mention that people are also interested in clinical microbiome studies. So we know that the microbiome can affect clinical outcomes. And so now people are directly using the microbiome in the clinic, all right, where they're trying to modify the microbiome over here. This can include things like probiotics, prebiotics, or even fecal transplantation in this case. So even though you don't know the mechanisms, people are still doing a lot of these types of studies. Fecal transplantation, you're taking poo from one person and giving it to another person, or these days, you can even get this in capsule form. What are some of the opportunities over here? We know that not all bacterial products and poop donors are the same. So one interesting subject would be trying to identify which donors are best for you given your current microbiome profile. Very limited work on that. People are interested in creating artificial poop cocktails or probiotic products over here. Again, the statistics for trying to figure out which combinations of taxa should be used here are extremely, extremely underdeveloped. And finally, all right, as with all clinical trial stuff, we're always looking to see if there's other designs that are more appropriate for this treatment modality as compared to others. So I'm just going to conclude with a couple of remarks over here. I think the microbiome is a field with a lot of potential to make impacts on human health, and also a field that can allow a lot of data scientists and statisticians to truly do impactful science. Making impactful contributions, though, is hard. All right. It's hard to change established approaches. All right. It's hard to it's really hard to work within fields that are already very well developed. So my suggestion, find areas that are underdeveloped, nascent, or are very chaotic right now. Ultimately, your approaches have to be practical. They have to work on real data. And really, you have to get new insights over here. Area that a lot of people are talking about is generative AI. This is something that I see people trying to do in the microbiome, but I've not seen any truly compelling work yet. All right, and I will stop here. Thank you.
Thank you, Dr. Wu. I think we have some comments and questions on the chat. Let's see.
All right, so should I go through it over here or? All right, just starting at the top. Yeah, the abundance. So right, as Monty McGee said, the abundance is considered proportion of a whole. That's due to the transformations that we use. And so methods for compositional data are highly applicable. As I've said over here, that I simply have not seen it truly make a big difference in a lot of data analyses. But it is still something that that many investigators are very concerned with over here. And so hence a lot of the tools try to take advantage of this. But as I've also said, a lot of other types of data are inherently compositional as well, including the early DNA microarrays and metabolomic studies, proteomic studies. We just normalized by the total abundance over here. But nobody really does anything or really cares about seems to really care about that in this case. All right. But we can always disagree on this. But again, just from my experience, I haven't really seen it make too much of a difference. Next question over here, we see a lot of papers report results at different taxonomy ranks over here. Do you believe this could be yet another source of FDR inflation, assuming that people run differential abundance analyses at different ranks independently? Are there more principled alternatives over here? So this is a really interesting question. Oftentimes people will run analyses so you have data at the species level, but oftentimes people will run it at a higher taxonomic level because it decreases sparsity and in principle can boost power over here. And so within the field, there's people who I consider lumpers, where they always like lumping bacteria together, or there's splitters, people who always like analyzing each subcategory separately. It tends to sort of be a question of practicality, however, in terms of just, well, if you only have a taxon observed in a single person, you're never going to have enough power to identify any associations. So what is the right way to do it? I don't know. But does this potentially lead to type 1 error inflation? If you're not being honest about it, absolutely. You can potentially lead to type 1 error inflation if you're not adjusting for the fact that you're looking at different taxonomic levels. Now, adjustment for type 1 error and for false discovery rate and family-wise error rate can be challenging because a lot of the hypotheses are inherently nested. Once you start talking about nested hypotheses over here, how many tests you need to control for or which ones you actually need to control for, it's not always immediately obvious. People have developed strategies, including various false discovery rates, that try to accommodate this hierarchy or try to do hierarchical testing in more principled fashions. But again, this is not something that but I'm not sure that there's any approaches that are truly satisfactory at the end of the day. That being said, is this an area where I'd put a lot of attention? Honestly speaking, you could from a statistical standpoint, but in terms of impact, I'm not sure that these are approaches that people will truly end up using that much. It is of statistical interest, but not really something that you can do a lot. But I think it will still be hard to convince people to use those tools over here. So Lee said, in the HIV study example, what R squared for HIV status can individual studies or a smaller subset of studies achieve? All right, hope to get a sense of whether or not pooling data actually helped at all. I actually don't have the numbers for that within the context of that study by itself. And so I don't have a good, I'm not able to really say. However, in other studies that we have worked on where we've pulled data, all right, oftentimes you do gain something by boosting sample sizes over there. But it really is study specific and contextually specific. Sometimes if things are too heterogeneous, then you're just contaminating good studies by pooling in weaker studies simultaneously over here. Okay. A lot asked about sample sizes for microbiome data, particularly within the context of longitudinal analyses. Sample sizes vary tremendously within the context of microbiome. There are some studies where you have very, very dense sampling on just a few patients, but then there's also studies where you may only have 2 time points collected on thousands of patients or subjects over here. It really depends on the particular study, and I don't know that there are any specific rules for what people have determined in this case. It really is just a question of practicality. A lot of longitudinal studies that I've personally participated in are within the context of clinical studies, where oftentimes the sample size is dictated based on the sample size of the clinical trial, and it's not based on the microbiome component directly over here. Final question over here from Gang. Do you have any suggestions to reduce cage effect for microbiome data analysis in mouse? So this is a very interesting and extremely challenging problem. The issue is that mice that are caged together tend to have very similar microbiome profiles. The reasons for this, there's a lot of them. 1, similar environments. 2, similar feed. 3rd, they eat each other's poop over there. All right. And so when you eat each other's poop, all right, it ends up that there's a lot of things that are transferred in this case. In fact, that's sometimes how they first identified microbiome effects. How to truly reduce that, I'm not entirely certain in this case. I think it's simply something that you're going to have to control for in your analyses rather than scrub out. But how to actually deal with it, I'm not sure in terms of cleaning your data. It's more something that, again, I think you're just going to have to accommodate in some way. All right. So I think that's it in terms of questions. If you have any other questions, I'm happy to take them offline via email or otherwise discuss. All right. Thank you so much.
Beautiful talk, Michael. It's awesome.
All right.
Thank you.
All right. Thank you.
Thank
Thanks,
you.
everyone.